I want to talk a little bit about apheresis material for autologous cell therapy and its subject to variability related to its limited quantity. The difference between patients and the differences across apheresis facilities. There's quite a bit of variability there. Omkar, I'd like to start with you, and then I'd like to ask Donna the same question. Talk us through some of the ways in which variability can be reduced for apheresis material for an autologous cell therapy.
Yeah. Again, going back to the point I made earlier, one of my colleagues, Jennifer Raven, who's an oncology nurse, we actually had this discussion not too long ago. If you think about apheresis, like the one thing to do is trying to harmonize as much of the apheresis procedure as possible. So you can have, you can have some degree of standardization in the way these cells are collected, processed, and then shipped to a manufacturer, right? But the other thing, which has not been put into place yet, and we're really pushing hard for the industry to adopt this approach, is that if you think of apheresis material today, to my knowledge, there are two main companies that own majority of the share of the market, right? They make two machines, right? And one of them is a clear majority over the other. So think of all the data that one company and the one company's product collects over a period of time. It puts that specific machine or that specific platform in a very unique position where you have this tons of data. You can do a whole sort of like whole list of like analytical the data analytics, I would say, on that data to really identify, how you can reduce this apheresis material variability. And we can talk a little bit again as an example of such a data analytical algorithm that we've been working on. But that's the reality of it. So why are we, with 6 years, no, to 2017. So we are 7 years since the first FDA approved CAR T, right? And why is this not being adopted yet or explored yet? And maybe it is, and I'm not aware of, but that's where I would challenge us as an industry to really think about what we can do to reduce that first step, collection of that raw material.
Okay, thank you. Donna, I would like to ask you the same question. If you could talk us through some of the ways in which variability can be reduced for apheresis material for autologous cell therapy.
I like to throw in my favorite phrase is fit for purpose. And so I think you have to consider what you're doing, what your manufacturing process, and what your desired product outcome is. That being said, one of the lessons learned very, very early on, as Omkar mentioned, there's two leading devices for apheresis out there. And one company is by far used more than the other. However, over the course of time, those institutions, along with those companies, have well-established methods for collecting leukapheresis over generic apheresis products. And from my perspective, it's best to stick with that lesson learned the hard way. And in talking with groups like the NMDP and the Be the Match type people, one of their QA people hit me up in the face, said, Donna, every time you tell me you want this done instead of that done, it will be a deviation. It will be a variance, because these companies go through, these institutions, these hospitals do this day in, day out by the established methodologies. And when a company comes and says, but okay, for our patient, we want you to do this, this, and this, they're more than likely going to not get it right. There will be a variation because they don't do it all the time. It may come up once every 3 months, and then here's another company that has a different approach. So I found that it's best to stick with the established protocols and know what that material is going to look like when it comes in. Know what we expect the outcome to be. And then if we don't get that, that's where we internally make different courses of the action that we have well established with the agencies. If we see this, then we're going to do that. If we see immature granulocytes, we're going to do this. Immature reds, we're going to do this. If we have patients coming with immature platelets, we're definitely going to process X, Y, and Z way. We've established all of that to give us the cleanest product at the end to go into manufacturing. One of the things that I think is important is to consider, I think bringing in your mechanism of action becomes important. There is the carte blanche to follow traditional blood banking practices of everything gets frozen. Well, garbage frozen also just garbage at yield. And there's some material components of the leukapheresis that can interfere. And I used the example of immature grands. They do not freeze and thaw, and you get sticky, snotty DNA when you thaw them that can interfere with your target cells. So consideration of how you want that to receive and ship and receive that product is important. I'm an advocate of room temperature collections because it maintains the integrity of the T cell functionality, and that's what our program is about. So we room temperature ship. Yes, that presents its own challenges because we have to have that material, again, through well-established studies, that we need that material to maintain its integrity received within 40 hours of collection. So that gets back into supply chain logistics that have to be managed and dealt with. There are solutions for that. So that's what works for us. That's not going to be necessarily what's for anyone else's given product, but it goes back to understanding the fit for purpose. What do you need that product to be, and what do you need to do to make it what you desire it to be? Maybe sometimes there's unique enough that you do need to go to the apheresis center and have them do something different. They can make modifications in those collection process, but I found it far better to stick with the industry standards that have been well established in the blood banking industries.